Okay, so let's begin our lecture on neoplasia, or you can also say cellular proliferation, cancer. So let's have a look at the cellular level review. Throughout our lifetime, most body cells are undergoing division, reproducing at a rate that is unique to that tissue type. So we have already discussed this lecture in the class, but this is just a review. So what does this line say? Most of that our body cells, all of them, undergo reproduction. Okay, not except the nerves and the muscle cells. Most of the cells undergo reproduction. Now they reproduce at a rate which is unique to the tissue, right? So as we discussed, skin, like our skin tissue, it keeps on replacing itself and it keeps on reproducing at a faster rate, whereas the cells of the liver or some other body part might not require replacement at such a high speed. So they reproduce at a slower rate. They only reproduce if some part of the liver is injured, or if it has been if you donate a liver part to somebody else, then only a need for the liver cells to reproduce arises, right? So they do not reproduce so fast as our skin cells. So we shed our skin so much, so yeah, skin cells reproduce at a faster rate. After division, daughter cells should resemble parent cell. Okay, this is like every cell, of course. Like if a skin cell reproduces or divides, it should look like and behave like a skin cell. It should not look or behave like a liver cell, right? So after division, daughter cells should resemble parent cell, and the DNA material and everything should be identical. The process of cell division generally follows a cell cycle. We know about that. We have done that in our first lecture also, right? So what are the phases of cell cycle? G1, S, G2 and M. When a cell is not dividing, it is generally in a G0 phase, the resting phase. Okay, and here this image was included to tell you or to remind you that there are cops of the cell cycle, and they are called as cyclins. Okay, there is cyclin D, E, A and B, so like alphabetically A, B, D and E. Okay, cyclin, they all keep a check uh, on the process of cell division, on the cell cycle. Okay, what are their main rules? Their main rules is to make sure that the DNA material is okay, the cell that is reproducing is okay, there is nothing wrong with the cell. Now this was included to make sure you understand what are the two proteins. Okay, so there is RB protein, or also called as retinoblastoma protein, and P53 protein. These are the two which are called as anti-tumor genes, or tumor suppressor genes. So what is the main rule? If they are like, they are even head of I don't know what they are called, but they are even head of the cows. Okay, so like cyclins detect a problem, that okay, something is wrong, then they will take the wrong cell to these proteins, that okay, RB protein, do something. So what they will do, they are the ones who will pass the judgment. They will either say that the cell is repairable, or the cell should undergo apoptosis. Okay, there are other oncogenes also, like BRCA1, BCL2, that it is just to show that these two genes act at these two checkpoints. These two checkpoints are made useless by these oncogenes, and so the cell can undergo neoplasia. Okay, I just picked it up from reference, so don't mind about these oncogenes. It is just to remind you that there is an RB gene which acts from G1 to S phase, and there is a P53 gene which acts in the S phase. Okay, now again, coming back to the basics of cellular review, muscle and nerve cells do not reproduce, but they have a very long lifespan. Epithelial cells reproduce rapidly, like example the skin, the lining of the organs. Okay, not the main on the not the tissue, like the lining. Like when we say lining, the lining of our esophagus, the lining of our stomach, of course they undergo reproduction, because every time we eat some spicy food, we kill some cells of the lining, some lining cells of the esophagus, right? Because the spicy food harms it, it erodes it. So we need those lining cells to reproduce, so even they cells reproduce rapidly. Then there are other cells that reproduce according to the need of the organ. Example, liver cells, which are called as hepatocytes. Okay, cell division requires oxygen and nutrients and adequate blood supply. So this is a very important line. What they are trying to say over here is there are two main things which a cell requires when it is undergoing reproduction. First of all, oxygen, and second is nutrients. Okay, both of them will be provided by a proper blood supply. Age will eventually alter the structure of a cell and produce death. Of course, after some time every cell undergoes aging. Okay, changes to DNA can cause cell death or alter in structure and function that can lead to mutation. Okay, any change in the DNA, either the change, if the cell survives, then it is said to have undergone mutation, but if it does not survive, it is definitely going to die. Okay, what are the other factors that can cause changes in the cell? There is chemical exposure, there are viruses, there is radiation, and there is trauma. All these factors can cause changes in the cell, like in the DNA material also. Okay, so like viruses like EBV and viruses like hepatitis B, all these can cause changes. HIV, these and all cause DNA changes. And radiation, of course, we know can cause DNA changes. Any trauma or chemical exposure can also cause problems to the cell. These factors influences increase chance of chromosomal changes and alteration in the cell enzymes and components. Okay, so what it means is that the chromosomes can change, and because of the change, either that or because of the change in the amino acid sequence, the proteins or the enzymes that are getting formed may get changed. Okay, when cells are mutant, function changes and normal cell cycle is lost. Okay, most defective cells die and are destroyed by immune system. We know that, right? Like most of them die, they cannot survive, and they are destroyed by the immune system by the process of apoptosis, phagocytosis, and all those things. Okay, what are the cancer characteristics and terminology? So cancer comes from a Greek word which means crab, literally said carcinoma, which was used by Hippocrates, the famous scientist, to describe the appendage like projections extending from tumors. So like a crab has, its appendages, its hands, like that cancer is also said that it has extensions like that, a cancer. So here when they are speaking cancer, cancer is generally used for malignant, those tumors that spread, those malignant tumors. Okay, when tumor is used for any kind of swelling, benign or malignant, any. But when we say cancer, it is used for malignancy. Okay, neoplasm means a new growth, any abnormal new growth which is following uncontrolled proliferation and serving no physiological purpose. So any growth, it can be a benign growth also, and it can be a malignant growth also. Any growth is called as a neoplasm. Cancer is a term which is used for malignant tumor. I just said that. Now what is the tumor classification and nomenclature? So the classification first we discuss is a benign and malignant tumors. What are the benign tumors? They are generally small, they are slow growing, and they are non-invasive. What do you mean by non-invasive? They are restricted. Like for example, this is a normal organ, okay? A normal duct. You see, these are the epithelial cells, and this is the basement membrane, the yellow colored. Here you can see. Now here, benign tumor, what you can you see the abnormal cells, the pink color cells are the abnormal cells, and you see they are within the limit, right? So they are generally small, slow growing, and non-invasive. Okay, they do not invade, they are well differentiated, and they stay localized. So they cannot invade and they cannot metastasize, they cannot go to different places. Okay, malignant tumors on the other hand are large. Do you look here? The size is bigger, they are fast growing, and they invade. Okay, they invade poorly, they are poorly differentiated. When I say poorly differentiated means these cells will not look like the normal cells. Okay, so poorly differentiated, and they can metastasize. So the important differentiating point between benign and malignant is malignant can metastasize, benign will stay localized, right? So they metastasize, infiltrate, invade, destroy surrounding tissue, then metastasize to other parts of the organ. So yes, malignant tumors are generally said to be fast growing, but there is an exception when we are talking about the tumors of the GI tract. Okay, so when we're talking about the tumors of the GI tract, they generally, yes, they spread fast, but they are detected at very later stage. Okay, they are detected like at very later stage. And benign tumors, if you look at the tumors of the ovary, the cystic tumors of the ovary, yes, it is, most of them are said to be small, but the benign tumors, benign cystic tumors of the ovary are really big in size. Okay, really big in size. I have seen 7 kilogram weighing benign cystic tumor of an ovary. So it's not, it is not like it's not like all the benign tumors always small. Okay, it's like, but general, this is like the general features. Generally benign are supposed to be small and malignant are supposed to be large. But the most important point that we have to always remember is benign will always stay localized, it will not metastasize, whereas the malignant, it metastasizes. Okay, now what is the definition of a cancer? Disease in which the abnormal cells divide without control and are able to invade other tissues. Okay, I said right, cancer is always used for a malignant tumor. So if you can use this definition for a cancer as well as a malignant tumor, so it is a tumor where the abnormal cells are dividing without control and they are also able to invade other tissues. Now, the first classification was depending on benign and malignant. The second classification is according to the tissue of the origin. Uh, Again, now it was generally said that benign tumors have a suffix of oma, lipoma, leiomyoma, whereas the malignant ones will have a suffix of carcinoma or sarcoma. Okay, but here again, oma exception is melanoma and glioma. I'll tell you why. Because when it was newly found out, newly identified, uh, when the melanoma and glioma were newly identified, this melanoma was a skin tumor and glioma was a tumor of the brain, so they thought it is a benign tumor, so they gave the names of melanoma and glioma. But later, as they started studying the features and they started studying even more detailed study, they came to know that, oh, they are like really bad malignant tumors. Okay, now, malignant tumors will have generally carcinoma and sarcoma. Carcinoma tumors are the ones which arise from the epithelial tissue, like the squamous cell carcinoma, adenocarcinoma, and sarcoma is said to arise from the connective tissue. So, sarcoma is liposarcoma, leiomyosarcoma. Now, sarcomas are generally less common, meaning they are not really, they are not seen at such a frequency as the epithelial tumors. Okay, but they are less common, but they spread rapidly. That is, they can, because they are malignant, they can go to other parts of the body quite rapidly, and they are highly malignant. Okay, they are like really malignant. But the other thing is, it's very difficult to how do you say, it is very difficult to identify. These tumors are very difficult to identify, because they grow at a very slow rate. Okay, they are very malignant, but they grow at a very slow rate. So because the surrounding tissues keep on adjusting to their size, they get identified at a very late stage. Like, you realize that there is a sarcoma present when it has spread a lot. Okay, so that is about sarcoma. What are the examples? You notice when lipoma is a benign tumor of the fat, whereas liposarcoma is a malignant tumor of the fat. Okay, now leiomyoma is a benign tumor of the smooth muscle cells, whereas leiomyosarcoma is a malignant tumor of the smooth muscle cells of uterus. Okay, again coming back to this diagram, just to remind you about the different changes that the cells undergo. So cells can undergo atrophy, as you see over here, no longer functioning, or they can go hypertrophy, bigger in size, or they can go hyperplastic, more number in size, or they can form metaplasia. These are the normal cells, tall columnar you can see, and they have been replaced by cuboidal cells. You can see over here, different types entirely. Sorry. So that is metaplasia. Dysplasia is where these cells have lost their features, as you can see over here. These are the normal cells, and here look how they have become abnormal. And neoplasia is, as we know, they start coming out of the basement membrane. Okay. Somebody asked me a question, I will answer it again. Hyperplasia and hypertrophic, both these stages, sometimes yes, some benign tumor cells show these two features. Okay, they also can be hypertrophic cells and hyperplastic cells, so they can also be caused as benign tumors. Like example, benign prostate BPH, benign prostatic hyperplasia. So it's actually a benign tumor of the prostate, and there the prostate cells are undergoing hyperplasia, right? So yes, they are both can be also characteristic features of benign tumors. Metaplasia is as I said, it's a warning. Okay, it's not a clear cut sign of malignancy, but it's a warning. Dysplasia is like a clear cut sign that yes, they are going to definitely turn malignant. Okay, metaplasia is more like a warning that it may or may not. Okay, but dysplasia is like yes, it is definitely going to turn malignant, so you do something, treat something, remove the tissue. Okay, and neoplasia, we know it's malignancy. Malignant tumors can be in situ or invasive. So here you see, just trying to give you another classification. You see a cell with mutation, how it has grown in number, that is hyperplasia, then how it is losing all the characteristics and becoming dysplasia. Now here till here it is limited by the basement membrane. Here this is the basement membrane, the gray colored one, right? But it is limited, so that time we will call it a in situ cancer. Yes, dysplasia and in situ, it is very difficult to differentiate these two stages, but generally dysplasia is like you will still see some normal cells. Okay, there are like 80% abnormal and 20% normal cells. Okay, but in situ you 95 to 99% of the cells are malignant. Okay, all those features of malignant cells you will see, so hence you can label it in situ because it is still within the basement membrane. Now here you see it has invaded, it has crossed over the basement membrane, so it is called as invasive cancer over here. Okay, now coming to the biology of cancer cells. So what forms the basis of the cancer cells? It is a disease of cumulative genetic changes, mostly during aging. So what are they trying to say by this line is, it is not generally one genetic change. Okay, one cell underwent one genetic change and cancer developed. No, it doesn't happen like that. It's like one cell undergoes some mutation, which is later on which is passed on to other cells, and those other cells again grow in more number, and they again acquire more mutational characteristics, like more radiation or more some other injury or UV radiation or something like that, and they further undergo mutations, and henceforth as many as they keep on undergoing those mutations, they will all result in a cumulative or additive effect in the end, and that will lead to the formation of cancer. Okay, so genetic changes. Now what are the genetic changes? A is a mutational. What are the kind of mutational changes that can happen in the genetics? It can be a small scale, like a point mutation, only one or few nucleotide base pairs are changing, or it can be a chromosomal translocation. Remember we did the Robertsonian translocation and stuff like that. So that is chromosomal translocation, that is a large change in the chromosome structure. Or there can be a gene amplification, that is a repeated duplication of a region of chromosome. A specific region of chromosome keeps on getting duplicated. Okay, those kind of mutations can be seen. What are the other features that can be seen? There can be epigenetic mechanism. So when I say epigenetic mechanism, what I mean is there can be DNA methylation. So what does DNA methylation mean? DNA methylation means a methyl group can get added to the DNA. Okay, our DNA does not like any guest on it. Okay, so when a methyl group, a CH3 group from any place comes and attaches itself to the DNA, that is called, the procedure is called as, it underwent DNA methylation, and that means it is going to help in the process of malignancy. Okay, histone acetylation, that is the histone, the positive charge particle which is keeping both the strands of both the chromosomes together, both the negatively charged chromosomes together, it undergoes acetylation. So what happens is the histone protein acetyl group comes and attaches itself to the histone protein. That makes the histone protein lose its positive charge. So as it loses its positive charge, the two strands of DNA starts getting separated. They start getting loosened up, and that can lead to genetic changes. Okay. Altered expression of non-coding RNA. You remember the RNA splicing that we did with the third lecture, like the introns and the extrons. The introns are the useless things which roam around. Extrons are the ones which will stay in the RNA and will code for the protein substance. Those introns, those non-coding RNAs, they can, they were supposed to be cops, but they can also cause mutation. Okay, they can also go crazy and affect the uh, characteristics of the DNA. They can cause mutations. Okay, so that was about mutations and epigenetic mechanisms. Now, what are the types of mutations? There are two types of mutations. One of them is also called as driver mutations, the ones which drive the progression of cancer. So what do I mean is, there are some mutations which will keep on pushing the cell towards becoming malignant. Okay, whereas there are some which are called as passenger mutation. So what does the passenger mutation mean? It just means that they do not really do anything. They're just, just adding up, just sitting there and adding up. The driver mutations will be pushing and pushing, like they will push the cell and they will change all the features, and they will turn the cell into malignant, and then they will again push the cell to multiply so that even more malignant cells are coming. Those are driver mutations. And the passengers will just be going along. Okay, you want me to stay here, I'll stay here. You want me to go into the baby cells, I'll go into the baby cells, stuff like that. Okay, that's passenger mutation. Now, driver mutation keeps pushing cells to become cancerous. Cancerous cells accumulate faster than normal cells. They form clone, also called clonal proliferation or clonal expansion. So as I said, what happens is mutations happened in the cell. Okay, it happened over some time, and then the cell turned malignant. So the mutation, it had mutation, it turned malignant. So as soon as it turned malignant, now it will pass on the malignant features and the mutation to its daughter cells, and so on. They will keep on multiplying because there are no cops in these cells, right? And there are nothing to regulate them that don't multiply or anything, so they multiply at even a faster rate. And with their fast speed and their with their wrong qualities, they all get together and they form malignant cells, a family of malignant cells, which is also called as a clone in the term basis. So that clone is called as a clonal proliferation or clonal expansion. So one cell gets into divides and becomes two cells, which further divides and becomes four cells, which further divides and becomes 16 cells, because each of them keep on reproducing along with the basic cell, right? So the basic cell is also reproducing, the daughter cell is also reproducing, the granddaughter cells is also reproducing, the whole keeps on reproducing and forms a clones. That is called as clonal proliferation, and that is how the clonal expansion leads to the malignant transformation in organ. That is how they keep on accumulating, and the organ can become malignant. Okay, just what I said. You see over here, the first mutation and the second mutation and the third mutation happens, and fourth or later mutation, the cell turned malignant, right? So here there was just injury happening, and now that so many mutations have happened and it has all got together, so it become malignant. Yes, I noticed that there are some genetic language over here. I'm sorry, just ignore it. Okay, it's just too much, so we are not going into so much of details, but this is just to show you the image. Okay, now what are the hallmarks of cancer? So when I say hallmarks of cancer, is every cancer has its unique features, unique characteristics. So what are the unique characteristics of cancer? So they are tumor promoting inflammation. Now this line is a dual meaning. Okay, the tumor promotes inflammation and the inflammation also promotes tumor. Okay, they both are like, okay, they both are promoting each other, kind of a thing. Then there is activating invasion and metastasis. Of course, a malignant tumor needs metastasis and invasion, right? There is genomic instability. There will be, yes, otherwise if it is stable, why would it turn malignant, right? So there is genomic instability. It is inducing angiogenesis. So what is angiogenesis? New blood vessels will be formed. Do you know why new blood vessels will be formed? Because the tumor cells require more oxygen and more blood supply and more nutrients, right? It will be resisting cell death, of course. Otherwise, if they would be dying, then why would the cell turn malignant, right? So it is resisting cell death. Deregulating cellular energetics. So what does it mean? The energy forming balance, the cycle, how to use up the nutrients and everything, it becomes deregulated. Sustaining proliferative signaling. What I mean by that is there will be a continuous signal to the cell to keep on proliferating. So as I said, the cell is reproducing, the daughter cell is reproducing, even the granddaughter cell is reproducing, all the cells are reproducing. They're constantly undergoing proliferation. Avoiding immune destruction. So they do not get destroyed by the immune system. They evade the growth suppressors. So they said there are some growth suppressors, some genes or proteins which suppress the growth. Okay, they do not let the cells to grow, but tumor cells find a way to escape them. Enabling replicative immortality. So what does that mean is, for the cell to be able to multiply for forever and ever, it requires immortality, right? Like for us to live forever, we require the never aging things, right? The immortality solution. So that is what the tumor cells acquire. They acquire the ability to replicate immortality. Okay, they cannot die. Now we'll look at all these hallmarks one by one. So here the hallmarks have been put into broad categories. The genomic ones. What are the ones included in genomics? Sustained proliferative signaling, evading growth suppressors, genomic instability, enabling replication immortality. Then there is cellular adaptation, how the cells adapt by inducing angiogenesis and by reprogramming the energy metabolism. The third category is resistance to destruction. How does it become resistant to destruction? By resisting the apoptotic cell death, that is the programmed cell death, by the tumor promoting inflammation, and by evading immune destruction. And the fourth feature is activating invasion and metastasis. Now all of these features we are going to look one by one quickly. Okay, so genomic, I have already explained. Sustained proliferative signaling. Normally cells express a growth factor, okay, which is like epidermal cells will express epidermal growth factor, so EGF. Now what does EGF do? The epidermal growth factor binds together two EGFRs, that is two receptors. The pathway that it uses is a PI3K pathway or MAPK pathway, and hence by either of these pathway the cell proliferates. Stay with me, don't be confused, please just stay with me. So what I'm trying to say over here is cells have receptors, okay, the receptors on which the growth factors are acting. The growth factors combine two receptors and they activate the cell growth by these two pathways, by PI3K phosphoinositol 3 kinase pathway or MAPK mitogen activated protein kinase pathway. Okay, I'm just telling you the name of the pathway, and as a result of the activation of this pathways the cell proliferates. What I mean is, as a result of this cellular pathway the cell will divide. Okay, now abnormality can happen where? Either in the growth factor receptor or in the pathways. Because of the abnormality either in the receptor or in the pathway, there will be a continuous signal for the cell to proliferate, right? So if you want a cell to divide continuously, you either mess with the receptor or you mess with the pathway. Okay, and if you have messed with any of these, the cell is going to get a continuous signal to keep on multiplying, to keep on proliferating, right? Now why do we need to know this basic? So what they did is, what scientists did it, they realized that okay, if you develop a drug which acts against these receptors, then you can stop the cancer, right? So that is what they did in the breast cancer. If you look at any breast cancer, there is ER report, that is estrogen receptor, PR progesterone receptor, and HER2 receptor. So the HER2 receptor is an epidermal growth factor receptor on the breast tumor cells, on the breast cancer cells. If the HER2 receptor is positive, they give the drug which acts against the HER2 receptor, and hence the cancer will not spread. The the signal to proliferate will stop, right? So that is why these growth factor pathways are important for the scientists, so that they could develop a targeted drug therapy for the cancer treatment. Okay. Next is evading growth suppressors. Now, normally you understand the term, right? Evading growth suppressors means they escape. They escape the growth suppressors. So, all those genes which are going to suppress the growth, which are going to suppress the proliferation, they evade, they escape those genes. Now, how do they do that? Normally, tumor suppressor genes or anti-oncogenes work in the cell cycle. They prevent the damaged cells from proliferating. There are two copies, one from each parent of such anti-oncogenes. So both have to be inactivated. So two mutations are necessary. Okay, we just did in the cell cycle. Remember, we had those P53 and PRB genes. Those were tumor suppressor genes. Now these genes are always acquired from both mother and father both, and for them to stop working, both of them have to be stopped, both of them have to undergo mutations. Okay, so some tumor cells acquire those capability, and they can suppress both of them, both the genes, RB genes on the chromosome. So RB gene is mutated, and because of that, the malignant cells which were not supposed to proliferate, they keep on proliferating, and they can cause tumors like retinoblastoma or lung, breast, or bone cancer or anything. Okay, now what did the scientists do by knowing this information? They helped in targeting cancer screening to facilitate early cancer detection and therapy. So if we know that okay, their RB gene was mutated, like they have a family history of retinoblastoma, so what they do is they keep a screening. Similarly, there is like an APC gene for colon cancer. So if you know that your family member, somebody in the family had colon cancer, so you can get yourself screened if you have that abnormal APC gene or not. If you have that abnormal APC gene, you are going to develop colon cancer. But if you do not have that abnormal APC gene, then you are not going to develop colon cancer. So these tumor cancer genes can be screened, and as you screen them, you can predict if the cancer is going to develop or not. Okay. Genomic instability. There is increased tendency of alterations, mutability in the genome during the life cycle of cells. Inherited or acquired mutation in caretaker gene like TP53 or P53, same thing, checkpoint gene, results in increased level of genomic instability and risk for developing cancer. So we just saw an example for PRB, the retinoblastoma protein. This is for the protein 53, P53, also called as TP53. Okay, so it is supposed to be a caretaker gene, but if some problem occurs in the caretaker gene itself, then of course the person is going to develop cancer, right? So example is xeroderma pigmentosa, where how does the problem happen? There is a defect in the repair of DNA which is caused by the UV light, the ultraviolet rays of the sun, and therefore it increases the risk of skin cancer. Okay, that's why we say that whenever you go out, protect yourself from the sunlight and from the UV rays and everything, because those UV rays give mutations to our DNA, and there are problems, and it can lead to cancer, skin cancer like conditions. Okay, enabling replication immortality. Normally, telomeres placed at chromosomal ends by telomerase enzyme. These telomeres shorten with division, and at the end to cease signal cell division. Cancer cells activate telomerase to restore and maintain telomeres, thereby allowing continuous cell division. Telomerase activity can be altered by loss of function of tumor suppressor molecules p53 and Rb. So what they're trying to say is chromosomes have two structures, okay, two strands. At the end of the two strands, there are telomeres. The ends of the chromosome, there are telomeres, which are placed there by the telomerase enzyme. Now, those telomeres, they keep on coming to the end of the chromosome, and the chromosome can keep on undergoing replication. It can keep on dividing. So there is one point when the telomere will stop coming at the end of the chromosome, and there is when the cell will stop dividing. What did the tumor cells do? They made sure that telomere was always present at the end of chromosome. Okay, so the chromosome always kept on multiplying and it always kept on replicating. So that is how they acquired immortality. They use the telomerase enzyme. So the telomere keeps on going to that place and the cell keeps on getting, the DNA keeps on getting replicated. That was all about the genomes. Now coming to the cellular adaptations, inducing angiogenesis. Tumor cells, like the cells of inflammation, require oxygen and nutrients for growth that is provided by the blood supply. So we know, right, they require oxygen and they require nutrients. New vascularization or angiogenesis, formation of blood vessels, ability developed by the tumor cells to continue with multiplication. So what they're saying is, for the tumor cells to multiply continuously, they kept on making new blood vessels, also called as new vascularization or angiogenesis. As they kept on making new vessels, the tumor cells kept on dividing because they got their supply of oxygen and the nutrients and everything, right? Reprogramming energy metabolism. So what do, because they require energy to metabolize, to divide, right? So they had to develop a way so that they could get energy continuously, and they could also get material to make new cells continuously. So normal cells in adequate oxygenation undergo oxidative phosphorylation in mitochondria, and they generate 36 molecules of ATP by breaking down each glucose molecule into H2 and CO2. Okay, normally the cells undergo oxidative phosphorylation in the mitochondria and 36 molecules of ATP are formed, and the end product is water and carbon dioxide, which we exhale out, right? In the absence of oxygen, normal cells perform glycolysis, that is anaerobic glycolysis, generating 2 molecules of ATP by breaking the glucose. Water, and the end products are lactic acid and pyruvate. Okay, now what did the cancer cells do? They are reprogrammed to use glycolysis, which is called as Warburg effect, even in the presence of adequate oxygen, and byproducts lactic acid and pyruvate used as building blocks for rapid cell growth. So why did they do that is, maybe the tumor cells are multiplying so fast that so much of oxygen cannot be made available to the tumor cells. Okay. Now if the oxygen is not made available, they undergo glycolysis. But what do the tumor cells do? We are not switching between the two processes, we will always undergo glycolysis. So they always undergo glycolysis, that is the breakdown of glucose, but the byproducts are formed at the end of that, and they are called as lactic acid and pyruvate. So what do you do with that? They are supposed to go out, they are supposed to be excreted, but they use those byproducts to form more cells, because these byproducts are used as the building block. If you look at carefully, pyruvate, it helps in those RNA DNA building block molecule. Okay, and lactic acid also helps in the formation of new cells. So these two are again used to form more cells. So the cancer cells are very smart. Not only do they get their energy molecules, they also do not require oxygen, and they can also get more building blocks. Okay, so you see over here just a diagrammatic image. It is not for your exam, but this is what a normal cell does. Pyruvate lactate oxidative phosphorylation 36 molecules of ATP, but if oxygen is not there enough, then the glucose goes and forms pyruvate and lactate, and 2 molecules of ATP are formed. What did tumor cells or the cancer cells do? It will have glucose which converts into pyruvate and either use O2 and get converted into CO2, or just release a lactate, and the 4 molecules are ATP are formed because it keeps on going as a cycle. It keeps on undergoing the process of glycolysis. Okay. Next portion was resistance to destruction. How do the tumor cells form the resistance? Resisting apoptotic cell death. We know apoptosis, right? Apoptosis is a programmed cell death, that is a mechanism by which an individual cell can self-destruct when needed. And what are the two pathways to activate apoptosis in a cell? There are two: intrinsic, extrinsic. Intrinsic gets activated by the stress, radiation, genomic instability, chemicals, anything that is inside. Okay, it activates the caspase 9 with the help of cytochrome C, which in turn activates caspase 3, which leads to cell death by cutting the DNA. Okay, again, please don't be scared by these terms. We just remember two: caspase 9 converting into caspase 3. Caspase 3 is active and the cell undergoes death by cutting its own DNA. That is the intrinsic pathway. How is it activated? By stress or radiation or genomic instability or chemicals and stuff like that. Now, what is the extrinsic pathway? The extrinsic pathway works by a receptor. And what is the name of the receptor? It is a fast ligand or CD95, which are present in the cytotoxic T lymphocytes or NK cells. They activate the caspase 8, which in turn activates the caspase 3, leading to cell death. Okay, so the basic thing we need to know is caspase 3 is present in both the pathways. The 9 is associated with the intrinsic one and the 8 is associated with the extrinsic one. The intrinsic one is started by the stress, radiation and genomic instability. The extrinsic one is activated by the receptors on the cell surface, the fast ligand and the CD95 receptors. Okay, and this is how the normally the apoptosis process undergoes. Okay, this is the normal process. Now what do the tumor cells do is they acquire way by which these apoptotic pathways are stopped. Okay, most commonly what they do is TP53 gene activates the defective cell death in the cell cycle by activating apoptosis. In some types of cancer, TP53 genes are mutated, there is a loss of function, apoptosis cannot be activated, leading to accumulation of abnormal cells. Some cancer cause downregulation of caspases or produce caspase inhibitors. So what they can do is they can either, so you know, right, RB gene and TP53 were the genes which were useful or which were the ones which started the process of apoptosis. So either they will stop those two genes or they can act against the caspase. Okay, the caspase 9 and 3 and 8, you remember, either they can work against those caspase. Okay, just today have a look at the diagram. It was the same. Okay, this is the extrinsic ligand caspase 8, caspase 3, that's substrate apoptosis, and this is the intrinsic one which is causing DNA damage. P53 is problematic, and so because of that there is caspase 9 which activates caspase 3 and the cell undergoes death. But if there is no P53 or if the P53 is defective over here, this thing will not go on, right? This process will not happen, so apoptosis will not happen, so the cell will keep on multiplying. Okay, now what are the other factors of resistance to destruction? Tumor promoting inflammation. I said chronic inflammation with H. pylori associated with peptic ulcer may lead to gastric carcinoma or gastric melanoma. Asbestosis silicosis causes chronic inflammation which can lead to tumor like mesothelioma, lung cancer. Hepatitis B and C is said to lead to hepatocellular carcinoma. These are just the examples. Okay, it is said that the H. pylori infection, which causes a chronic condition like peptic ulcer, can lead to gastric cancer. Foreign antigens like asbestos or silica in the lungs can cause chronic inflammation which keeps on injuring the lung, and it can cause a malignancy of the lung, causes mesothelioma or lung cancer. Hepatitis B and C are the viruses present in our liver cells, and they can cause hepatocellular carcinoma.